Delighted to introduce Dr. Michelle Wong as our seminar speaker today. Michelle has her bachelor's degrees in sociology and molecular environmental biology from UC Berkeley. After she finished her undergrad, she stayed as a lab and field technician at UC Berkeley in Wendy Silver's lab. And then she went on into government and was an environmental scientist at the California EPA Department of Pesticide Regulation. At that point, she realized she wanted to continue studying ecology, and she went to the Cornell University and did a PhD in the Department of Ecology and Evolutionary Biology. We were lucky to have Michelle join us as a postdoc at the Cary Institute in 2019, where she's a biogeochemist, an ecosystem ecologist, and a wonderful collaborator, colleague, and community member who studies how plants and microbes interact with nutrients to shape terrestrial ecosystems. And I am really excited to have her speak with us today. Welcome, Michelle.
For the introduction, Sarah. Can everyone hear me okay? Great. So yeah, like Sarah said, I've been at the Cary Institute since the fall of 2019. And today I'm going to share a little bit of the work that I did before I came to the Cary Institute and some of the stuff that we're working on with many of the scientists here at the Cary Institute. And like Sarah said, I'm a biogeochemist and an ecosystem ecologist, and I think a lot about how nutrients cycle in the world and how they support the function of terrestrial ecosystems. So this is a map of mean annual net primary production, or the net carbon that plants take up over time. And I think a lot about how nutrients support this production. So humans are emitting about 11 gigatons of carbon into the atmosphere every year, which causes climate change. And just for context, 2 gigatons of carbon is equivalent to about 1 ppm of CO2. But only 40% of this carbon that we emit actually stays in the atmosphere. The rest is taken up by oceans and by the land carbon sink. About a third of the carbon that we emit is taken up through increased rates of photosynthesis into terrestrial ecosystems. And a huge proportion of this land carbon sink is actually in temperate forests and then also in tropical forests. So in intact and in regrowing tropical forests, these really productive ecosystems just help mitigate climate change. But one of the biggest questions is really, what is the future going to look like? Are these plants going to continue to photosynthesize and take up carbon at the same rates? So this is a projection of an increase in biomass in response to 250 ppm of CO2. And as you can see, the strongest or highest projected biomass increases are in the tropics, but they're also highly uncertain. So we really don't know what these future ecosystems are going to look like. So a big question again is if the land sink is going to continue to take up carbon at the same rates indefinitely, or if eventually they will be limited by resources such as nutrients. And two of the most important and often limiting nutrients for terrestrial ecosystems are nitrogen and phosphorus. So nitrogen is required for nucleic acids, amino acids, and proteins such as rubisco, which is what drives photosynthesis. And phosphorus is required for also nucleic acids and then energy compounds such as ATP. And the reason why nutrients are so important is because the soil doesn't have an endless supply. That's why we use fertilizers to grow our food. And that's why the industrial production of fertilizers has allowed human populations to double in size. And with Earth system models, which are the most powerful tool we have to project the future, project the land carbon sink into the future, and then don't incorporate nutrients, this is the black line indicated here. Oh, maybe the mouse. Okay. And then when they incorporate nutrients, they find a decrease in net primary production by about 20%. So those red and the blue lines here indicate what the land carbon sink will look like in the future with nutrient cycling. And this is a map of projected nutrient limitation globally. So as you can see, northern latitudes, temperate regions and boreal regions are mostly nitrogen limited, and tropical regions are thought to be mostly phosphorus limited. And when this map was published in 2020, they addressed this really huge knowledge gap that we didn't have a global map of terrestrial nutrient limitation. But also a map like this doesn't account for changes across time, and it doesn't account for spatial heterogeneity at smaller scales. And also it doesn't account for the nutrient limitation of specific ecosystem processes. So the work that I'm going to be talking about today really is focused on understanding where, when, and to what extent are terrestrial ecosystems limited by nutrients. And to do that, I'll talk about 3 different questions and areas. The first of which is, how are nutrients distributed in the environment? So where can we expect nutrients to be limiting? The second is, how does nutrient availability change after disturbances? So disturbances are a huge part of our ecosystems, and that can tell us a little bit about when nutrients are limiting. And finally, how do plants address and overcome nutrient limitations? So we know to some extent that plants can interact with nutrient availability, and they don't necessarily just solely reflect what's going on in the soils. And I'd really like to acknowledge all of the collaborators and scientists and everyone involved in all the projects today, especially some of the folks here at the Cary Institute, and I'll talk a little bit about the work that we're doing at the end. So moving on to that first question, how are nutrients distributed in the environment? So we pretty much need to understand where nutrients come from. And for nitrogen, which composes 80% of our atmosphere, nitrogen is brought into the ecosystem through biological nitrogen fixation. And as you can see here, the warmer colors indicate higher projected global nitrogen fixation. And about 80% of global nitrogen fixation is thought to occur in the tropics. And there's 2 main pathways for nitrogen fixation, the first of which is symbiotic, and that occurs as a mutual relationship between specific tree species, often in the Fabaceae family, and then bacteria that form root nodules with these plants, and they can fix really high rates of nitrogen. So soybeans are a really powerful example. And the other pathway is free-living nitrogen fixation, and this occurs mostly as a heterotrophic process. So during decomposition of leaves and litter, you'll find it in soil and in litter. And the tropics are thought to have high nitrogen fixation rates because they have a high abundance of these nitrogen fixing trees. And because there's often a lot of warm and wet conditions that can facilitate a lot of decomposition and thus free living nitrogen fixation. But the process of nitrogen fixation actually has its own constraints. So molybdenum and phosphorus are 2 rock-derived nutrients that are thought to constrain nitrogen fixation. And the reason for that is because molybdenum is a cofactor in the nitrogenase enzyme, which catalyzes nitrogen fixation. And then phosphorus is required for ATP. And historically, it was thought that phosphorus was the main control on nitrogen fixation globally, but a previous study showed that molybdenum limitation could be more prevalent than previously thought. And this is really critical because molybdenum is already rare in bedrock material. So how rock-derived nutrients actually get into soils and into the environment is through the weathering of bedrock. And that eventually weathers into saprolite and then to the soils that you see walking around today. And molybdenum is already rare in bedrock. And when it's oxidized, it tends to be very soluble. So it leaches into rivers and then into oceans where it has one of the highest concentrations of trace metals. So it has this really interesting distribution where it's rare in soils, but it's really abundant in oceans, for example. And just to compare tropical regions with temperate regions are often have less wet and warm conditions that can facilitate high rates of weathering. So you have less access in tropical regions to that original bedrock material because these soils are so weathered that you've lost that, access to that original bedrock. So this leads to this really interesting question. Where do tropical forests get their molybdenum to support these theoretically high nitrogen fixation rates? And we had a few suspects. So we figured that because geological weathering or weathering from bedrock wouldn't be as critical in tropical forests, that they might be more sensitive to atmospheric inputs. Sea salts, like I had mentioned earlier, molybdenum has a really high concentration in seawater. And then volcanoes and dust can also emit aerosols into the atmosphere where they can get transported thousands of kilometers and then get deposited on land. And this may be news to you, but desert dust, especially from the Sahara, gets entrained into the atmosphere and then travels across the Atlantic Ocean and gets deposited across Central America and also the Amazon basin every year. So it's thought to contribute iron for phytoplankton in the Atlantic Ocean, and then also phosphorus to the Amazon. About 22,000 tons of phosphorus is thought to make its way annually over every year. And so my collaborators were able to collect samples from the Bow Delhi Depression. This is a photo of them collecting some samples. And that is a hotspot of dust production from the Sahara. And we were able to measure it for molybdenum and then estimate modeled deposition just to figure out where this molybdenum was going and where it was coming from. And so we used a three-dimensional transport model called the community atmospheric model. And then we focused on, again, dust, sea salts, and volcanoes, which are the dominant natural aerosols in the atmosphere. And then for sea salts, we used a molybdenum to salt ratio that's really well mixed in oceans. And then for volcanoes, we used a molybdenum to sulfur ratio. And what we found was that dust comprises about 70% of natural molybdenum deposition. So these warmer colors here indicate the source regions or higher deposition rates. And then the cooler colors represent lower deposition rates. So really, molybdenum deposition is not evenly distributed across the globe. And the triangles here indicate where we collected our samples from. And as you can see, it really does move across the Atlantic Ocean and then to Central America and then to Northern South America. And this is a map including sea salt spray and volcano emissions. So they make up about 30%. Sea salts are like 26%. And as you can see, especially in the Southern Hemisphere, with the dominant circulation patterns, a lot of sea salt spray does get deposited on the eastern side in the Southern Hemisphere. And this is and just to orient you, this region indicated by this blue arrow here is actually a rate that a previous study fertilized with. So even though these rates seem really low, in the milligrams of molybdenum per meter squared, just 1 year of deposition can potentially stimulate nitrogen fixation in some regions. And across time, so across tens to hundreds of years, this can really accumulate over time when you don't have a lot of inputs, again, coming in from bedrock. And this is a map including other nitrogen fixation studies in the tropics. And what I really wanted to orient you to was to the Amazon basin, which is supposed to be, supposedly thought to be a hotspot of global nitrogen fixation. And as you can see, the southern Amazon basin actually receives much lower molybdenum deposition than the northern Amazon region. And also there hasn't been, to our knowledge, nitrogen fixation studies there. And so I really wanted to characterize molybdenum in soils and see if it was going to affect nitrogen fixation in this region, in the southeastern Amazon. So is molybdenum low in the southeastern Amazon? So this is just to show you how weathered these soils are. This is a picture of me in 2 different soil pits that go to 10 meters, but the soils actually go deeper than 10 meters. So really, really weathered soil where you don't find any bedrock material. And then we did some concentrated acid digestions, and then these are some other molybdenum concentrations measured in other tropical studies. It's not like well characterized across Earth's surface, for example, but indeed we found that molybdenum in the southeastern Amazon was very low, like we hypothesized, just due to low atmospheric inputs and then low inputs from geological weathering. And phosphorus concentrations were also low, and the purple here indicates the average concentrations in bedrock across Earth's crust. So that leads to the second question: is nitrogen fixation then constrained by molybdenum in this region? And to do this, we conducted a long-term fertilization experiment across a year. And although a year may seem relatively short, previous studies focused on responses of free-living nitrogen fixation in 1 day, because bacteria can just turn over so quickly. And we did this with a randomized block experiment that we repeated in the wet and the dry seasons that were 2 by 2 meters squared. And this is a student Giovanna next to a lime plot. And the reason why we added lime in addition to molybdenum and phosphorus is because these soils are so acidic that they can actually bind onto these anions, making them less available. So we thought, okay, if we raise the soil acidity, we might expect that nitrogen fixation would increase. So that's this prediction that we have here. And surprisingly, we actually didn't find strong responses of nitrogen fixation. So in the top figure, I'm showing a response in the wet season in 1 week, and no significant responses of nitrogen fixation to these additions. But then 1 month, we did see a slight response in the soil to molybdenum, phosphorus, and lime. And as one does when you don't find strong responses, you take many, many measurements. I'm not going to show you all of these figures, but I'll walk you through a simplified version. So the columns here indicate the different treatments, and then the rows indicate the different time points that we measured responses to. And what we found was that there were really only strong responses in the soil at 2 time points. And this was really surprising because pretty much every other study that has measured nitrogen fixation in response to molybdenum or phosphorus in the tropics has found responses either to molybdenum or to phosphorus or to both. So why not in the southeastern Amazon? What makes this site different? And we hypothesized that maybe it was due to nitrogen availability. So here, just to put this back on the drawing board, on the x-axis, I have C to N ratio. And what a low C to N ratio indicates is high nitrogen availability. And then a higher C to N ratio indicates low nitrogen availability. So more carbon atoms, for example, to nitrogen atoms. And you would expect that free living nitrogen fixation would be low under high nitrogen conditions, just because it's going to be easier for bacteria to take up nitrogen from the environment than to fix it themselves. So you would expect some relationship like this. And what we found was that our sites in the southeastern Amazon were much more nitrogen rich than some of these other tropical sites. And so that means that even if molybdenum and phosphorus were made more available, you wouldn't necessarily see stimulation to nitrogen fixation because there's already so much nitrogen in the environment. And this is a figure showing surface inorganic nitrogen concentrations. And the concentrations just in the surface soils of nitrate and ammonium were just much higher or on the higher end than many other tropical regions. So really nitrogen-rich ecosystems. So in summary, we found that molybdenum deposition is probably really critical to some ecosystems, especially where there's not going to be a lot of inputs from bedrock. And that, like we expected or like we hypothesized, that molybdenum was indeed low just because of the low inputs from atmospheric and geological processes. But that nitrogen fixation was not constrained by molybdenum, which was really surprising. And that it would be really critical to, and that's probably due to nitrogen richness, and that'd be really critical to understand the drivers of variation across the tropics in the future. So going back to that original question, how are nutrients distributed in the environment? At least for rock-derived nutrients, I think we can utilize approaches looking at atmospheric inputs and geological inputs to tell us a little bit about where we would expect rock-derived nutrients to be low, but we really have to understand what's going on besides measuring total concentrations, because different dynamics can be going on. And I talked a little bit about where nutrients can be limiting, but next I'll talk about when. So how does nutrient availability change after disturbances? And this is really critical because a lot of nutrients are displaced after disturbance events, and we do find that ecosystems recover pretty quickly, but nitrogen is one example that's really locally controlled. And fire typically displaces nitrogen in a disproportionate amount relative to other elements because nitrogen is volatilized at a lower temperature and also is typically concentrated in leaf biomass, where basically it can get or fires volatilize nitrogen and they can blow away to other ecosystems, for example. Whereas phosphorus or calcium or magnesium tends to be more concentrated in wood biomass. It tends to get conserved as ash. So some phosphorus may be lost in the process from the ecosystem, but usually gets redeposited onto the forest floor. So in a previous study, in another forest fire in the Amazon, Rondonian, and Pará, Kaufman et al. Found that about 50 to 60% of the nitrogen was lost, but only 7 to 32% of the phosphorus was lost. So even though these systems are not necessarily thought to be nitrogen limited, they can be after a disturbance event like fire. And I'm going to zoom in on this map of Brazil. And this is a region indicated by the blue box between the Amazon and the Cerrado biomes. And it's a region where a lot of global tropical deforestation is occurring. Maybe you've heard of the arc of deforestation. And the reason that these forests are being cut is because they're being converted into pastures and croplands, indicated by the blue and the red here. And this has really big downstream consequences for the local water cycle, because about half of the precipitation in the Amazon is thought to come from trees transpiring water. So if you cut down these trees, they're no longer transpiring water and they become just much more dry. And when you combine these dry forests with ignition sources from agricultural fires, this can cause fires to spread where they haven't been before or they wouldn't normally spread to. So I think a lot of us remember when fires really made the news in 2019. These forests are really spreading, or fires are really spreading. And in drought years such as 2007 and 2010, about 12% and 5% of these southeastern Amazon forests burn. So it's really tragic. And my collaborators actually conducted a fire experiment starting in 2004 at Tengu Ranch in the southeastern Amazon. And this plot B 3 years indicates these plots that were burned, or one plot that was burned 3 times every 3 years. So 2004, 2007, and 2010. And the reason that they did this experiment was to be able to capture dynamics before and after these fires to really characterize what's going on. And these forests, or these plots are 50 hectare plots. They're relatively big plots. And then we also have a control forest on the right. So we have long-term data such as forest inventory data and then leaf area index measurements. And I focused on the top 10 most dominant species in the forest, which makes up about 50% of the total trees in both treatments. And because we have the long-term data, we're able to characterize some of the movement of nitrogen, for example. So we found that about half of the wood nitrogen was lost during these 3 low-intensity burn fires. And we're not seeing a recovery yet, but in the leaf nitrogen pool, about 75% of the nitrogen was lost, but we're starting to see a recovery at about 5 kilograms of nitrogen per hectare per year. And because we have this long-term data set, we're also able to characterize the trees that are recruiting into the ecosystem. So the basal area of potentially nitrogen fixing trees actually rapidly increased. And that's what you would expect, after a disturbance event, that these nitrogen fixing trees come in and they fix nitrogen. So about 1% basal area to about 10% in the burned forest. So it's really rapid growth of nitrogen fixing trees. And I wanted to measure nitrogen fixation at this time point when we're seeing this increase in tree abundances. And to do that, so asking, does nitrogen fixation increase to replace those nitrogen losses, especially that recovery that we're seeing in the leaf biomass? So to do that, we measured both symbiotic and free-living nitrogen fixation. And to measure symbiotic and fixation, we used 2 approaches, the first of which was a grid sampling approach. So sampling within a grid, just assuming that we don't necessarily know where trees are going to put their root nodules because they can spread their roots, meters and meters away. And then the other was to look at the base of the nitrogen fixing trees. And then for free-living nitrogen fixation, we measured it in the litter in the soil, again, where decomposition is thought to occur. And then we use the acetylene reduction assay, which measures nitrogenase activity. And then we paired that with a 15N2 incubation, so an isotopic tracer. And what we found was that symbiotic N fixation was slightly higher in the burn plots like we expected, but these rates were really, really low. So about 10 times lower than the pantropical estimate. And when we include free living nitrogen fixation in there, these rates are still less than 1 kilogram of nitrogen per hectare per year. So not really replacing those increases that we're seeing in the leaf biomass at about 5. And symbiotic or free living nitrogen fixation was actually higher in the control forest, likely due to a higher accumulation of litter fall, and then also more moist conditions that could facilitate higher free living nitrogen fixation. And I think the most surprising thing about this was just how incredibly low these nitrogen fixation rates were. Again, we typically think of tropical forests as fixing a lot of nitrogen. And just to compare to some other mature and secondary forests in the tropics where symbiotic N fixation has also been measured, these are really, really low rates in our study. So just vastly low rates. And the reason for that, we think, is that our group actually somewhat accidentally found these large pools of extractable nitrate just within the top 4 meters of those really weathered soils that I showed you before with the soil pits. And just this concentration alone, even if it's not necessarily plant available, is 2 orders of magnitude greater than the recovery rate that we're seeing. So even if some proportion of it was accessible to plants, it could explain why we're not seeing this rush of nitrogen fixation replacing those N losses. And the reason for that is because as soils weather more often, or they get more weathered, they typically lose their negative charge and they weather into 1-to-1 clays, which typically have more of a positive charge, and they're able to hold on to these anions like nitrate, whereas normally soils typically hold on to cations. And I think we are wondering, like, how common is this? Like, how underrepresented are some of these forests that we're studying? And so here I have mapped out in green broadleaf and deciduous tropical forests, and similar soil types to the ones that we had at our site in the southeastern Amazon. So oxasols that were topographically flat. And we find that about 40% of the tropics may be very similar in terms of just the topography and the soil type, so really weathered soils. And when we layer on previous nitrogen fixation studies, we find that there's not that many across these similar soil types. So if we were to do a thought experiment, for example, on like what this actually means for the nitrogen cycle, well, one, it means that nitrogen fixation rates may not be as high in some regions as we previously thought, but two, that maybe we've really perturbed the nitrogen cycle. So currently through fossil fuels and through agriculture, humans are thought to be adding about 100 teragrams of nitrogen into the environment every year. And if we make this assumption that tropical forests and savannas fix 80% of that, that means we've really doubled the nitrogen cycle. But if 16% of that nitrogen fixation may be lower, then maybe we've more than or closer to tripled nitrogen cycling in the environment. So really causing downstream consequences, biodiversity loss, and eutrophication. So in summary, fire causes large nitrogen losses, which is what we hypothesized. But at least at our site, nitrogen fixation did not increase to replace those losses. And in the future, I think we're seeing rapid recovery relatively in our sites, but it's unclear if more disturbances are going to cause a tipping point. So how does nutrient availability change after disturbances? I think it really is site dependent because in some ecosystems, you're going to see nitrogen limitation after a disturbance, but in others, it really depends on how much nitrogen is actually stored in the ecosystem that we're not necessarily accounting for. And I just summarized a lot of my work during my PhD, but I was really excited to come to the Cary Institute and start thinking about how plants actually interact with nutrient availability. And the reason for that is because just working in these really weathered tropical soils, I didn't show you any results from this, but Eric Davidson had led a fertilization experiment and they didn't find strong responses to phosphorus additions at our site in Tanguru, which is actually consistent with many fertilization studies across the tropics. There's not as strong, often strong as responses as one would expect, just based on how weathered the soils are and how low in nutrients they are. And to that point, I think it's because plants have this immense capability to actually obtain nutrients that we're not, or at least I wasn't thinking about originally. So how do plants address and overcome nutrient limitation? And more specifically, do plants adjust nutrient acquisition strategies in response to changing nutrient availability and changing plant demand? And traditionally, the way that I thought about nutrient limitation is that on low nutrient soils, you'd expect lower above ground productivity, so less biomass overall. But in reality, plants can use nutrients more efficiently. They can produce enzymes to access nutrients or find roots, and they can fix nitrogen and increase their symbioses with mycorrhizae. And the consequence of that may mean that above ground productivity, they're able to make up that difference and then eventually hit light saturation. So above ground productivity may be similar in less fertile sites versus more fertile sites, just because plants are maybe utilizing more strategies. And we wanted to test this across a secondary forest gradient. And the reason for that is because it really represents this change in plant demand. So are plants able to change their strategies in response to changing nutrient conditions, but also in response to this demand that they may need? And the reason for that is because harvesting processes like clear cutting, for example, take away a lot of nutrients just because you're harvesting that biomass. And then also nutrients such as nitrogen are just really mobile after you cut trees down. I think a lot of work at Hubbard Brook has shown this. That as trees are no longer taking up nitrogen, it tends to be lost into the ecosystem or leaches into rivers. And so these forests grow really rapidly early on in succession, and then they tend to just recycle their nutrients in the later fall immature forest. So you have much higher external demand early on in succession across forest age, and then less external demand because these plants don't necessarily need to mine soils as strongly as they did in young forests. And in addition to the change in the demand for nutrients, the change in nutrient limitation can change. And the reason for that is, or the type of nutrient limitation can change. And the reason for that is because phosphorus tends to stick around. It's not as mobile as nitrogen. So we could imagine that phosphorus availability is relatively consistent across forest age. But because nitrogen is lost easily after a disturbance event, it tends to be very limiting early on. But then nitrogen fixing trees may come in and they recruit in and then they fix nitrogen. So you get this shift from early nitrogen limitation to later phosphorus limitation. And you have this changing nutrient landscape where demand changes and also the type of nutrient limitation changes. So we wanted to test some of these strategies at the community level in response to this dynamic nutrient demand. I think we thought that it could tell us a little bit about, what plant strategies are going to look like into the future, for example. And one of these strategies is root phosphatase enzyme production. Bacteria can also produce these as well, but what it does is it catalyzes a hydrolysis and releases a phosphate atom from organic matter. So it's typically recycling or accessing organic phosphorus pools in the soil, and it allows plants to then take up that phosphate ion. And the other strategy that we wanted to look at was the utilization of mycorrhizae. So mycorrhizae are fungi that can trade photosynthetic carbon, and then they act as an extension of plant roots, and they can also modify the soil environment, so they can slightly acidify the soil environment, and they typically have higher uptake rates of nutrients than plant roots alone. But all of these strategies have a cost associated with them. So plants aren't going to necessarily utilize all these strategies unless they need to, right? Because phosphatase enzymes have a high nitrogen and potentially carbon cost, and then mycorrhizae, again, they're taking up they're utilizing photosynthetic carbon that the plants could otherwise allocate elsewhere. And so we wanted to test these strategies across this fertilization experiment that Sarah and Jeff Hall are PIs of in Agua Salud, Panama, and also in Gigante near BCI. And there were this is a really unique fertilization experiment in that it spans multiple forest age classes. So pasture plots, and then 10-year-old forests, 30-year-old forests, and then mature forests. And they were fertilized factorially by nitrogen phosphorus and then replicated across the landscape 4 or 5 times. And just when I was collecting these root samples from soils, the forests had already been fertilized for 4 years. So the 0-year forest was actually 4 years in succession, so 4-year-old forest. And this picture below is of Sarah in the 30-year-old forest, and you can just really see how much the forest has changed. It really looks like a mature forest in some capacity. And so we were able to collect roots at the community level from randomized soil cores. And then I'll just show you results for phosphomonoesterase today, which is the dominant phosphatase enzyme. And then we also stained the roots for mycorrhizal colonization. And what we hypothesized was that root phosphatase activity would increase across forest age because as these forests are shifting from nitrogen to later phosphorus limitation, there'd be a higher demand for root phosphatase as you go along succession, but that this would decrease again in the mature forests because there's less external demand for nutrients. And that nitrogen additions would stimulate root phosphatase production because there is nitrogen in these enzymes. And that phosphorus would suppress root phosphatase activity in the older forests because as phosphorus is added and made more available, then there'd be less of a reason for plants to upregulate this strategy. So more phosphorus limitation later on in succession. And these are the results. So I'm going to walk you through the results just for the control forests first. And the x-axis indicates forest age and then the different treatments. So control forests, nitrogen, phosphorus, and then nitrogen phosphorus. And as you can see, phosphatase activity did actually increase across secondary succession and then decreased again in the mature forests. And we found a stimulation of root phosphatase activity in response to nitrogen additions in the youngest 2 forest age classes, but then not in the oldest 2 forest age classes. And then we didn't find a suppression in response to phosphorus in the early youngest 2 forest age classes, but then we did see a suppression of phosphatase activity in response in the oldest 2 forest age classes. And so I will walk you through a simplified version of these results. Going back to our hypotheses, we did see this increase in root phosphatase activity across secondary succession and then a decrease again in the mature forest, as these forests shift from nitrogen to potentially later phosphorus limitation. And then we did see a stimulation by nitrogen early in succession, but not later in succession. And then we found a suppression, not early in succession, but then later in succession. So root phosphatase activity really behaved in a way that we really hypothesized. I think it's a really it's a nutrient strategy I think that we have a little more predictive capacity over. So next I'm going to talk about mycorrhizal colonization. And what we expected to find was that mycorrhizal colonization, so this is the presence of different mycorrhizal structures in the roots. And this index really captures if plants are investing more in mycorrhizae, we'd expect more of these mycorrhizal structures. And we'd expected mycorrhizal colonization to decrease across forest age as just nutrient demand changes and becomes less strong as forests get older. And we expected that maybe nutrients like nitrogen would stimulate mycorrhizal colonization early on in succession, just due to the fact that mycorrhizae themselves can also be nutrient limited, but that phosphorus would suppress mycorrhizal colonization, especially later on in succession. And what we found was really surprising, that mycorrhizal colonization actually stayed relatively consistently high across forest age. So these forests seem to be very, very mycorrhizal. And in the youngest forest age classes, we couldn't really see a stimulation just because they were already so colonized with structures, but that we start to see negative or suppression later on in succession, especially in the oldest forest age classes, we're seeing a suppression by mycorrhizae. So a simplified version of these results, we did not see this, we did not see a suppression of mycorrhizal colonization over time. We didn't see a stimulation either, but then we start to see a suppression later on in succession. So in summary, trees do seem to adjust root phosphatase activity in response to changing nutrient availability and demand, but we didn't see as consistent of patterns with mycorrhizal colonization alone. And I think it just speaks to this really complex nature, because symbioses with mycorrhizae can range from symbiotic, and that's how we think of it as plant ecologists, but it can also be parasitic as well. And I think it's just a difficult relationship to just untangle alone by measuring colonization. And this was really consistent with our meta-analysis. I'm not showing the results here, but we found really predictable responses in response to root phosphatase, but not with mycorrhizal colonization across some other tropical studies. But I think ultimately we really have to understand what the cost of these strategies are to the plant, because that's going to tell us a little bit about how plants are going to be able to upregulate these strategies in response to stronger nutrient limitation in the future. So how do plants address and overcome nutrient limitation? So we know that to some extent they can change these strategies, but I think one thing that we really need to think about is how communities can actually change on longer timescales in response to nutrient limitation. So I just showed you this figure earlier, and one thing that we can see in fertility gradients in the tropics is that you may not actually find the same species in different nutrient conditions. So you could start with, for example, the same seed bank in a high nutrient soil versus a low nutrient soil, but that maybe different species actually experience mortality and different species recruit in. So you have different species that specialize in different nutrient conditions. And the consequence of that may be that you don't actually see differences in above ground biomass between low fertility versus high fertility sites. And that's something that we're pursuing currently at the Cary Institute. And this work is still in progress, so I'm not going to show you all the results. This is a picture of a black locust field at the Cary, which I think that Mike and Fred maybe don't love to see. But the question here is if black locust accelerates phosphorus cycling in eastern deciduous temperate forests. So really alleviating phosphorus limitation at the community level, just through long-term changes in community composition. And what I wanted to point out here is that Robinia pseudacacia, or black locust, is the dominant symbiotic nitrogen fixing tree in eastern forests. It's thought to fix more than half of symbiotic nitrogen fixation in the US. And it interestingly has one of the highest growth responses to nitrogen deposition. And this is data from an FIA database from Horn et al. And what's really interesting is that you would actually expect nitrogen fixing trees to be less competitive and not do well under nitrogen deposition, because it's costly for them to fix N. So when N is in the environment, other trees should be able to capture that nitrogen and be more successful. But this is really the opposite of what we were expecting. And there's a few lines of evidence showing that these forests, eastern deciduous forests, which have been historically nitrogen limited, are maybe shifting towards co-limitation with phosphorus, or more phosphorus limitation in these forests, again, because of historical nitrogen deposition. So there's data from just the observation of foliar N to P concentrations in different forests in their leaves, and one fertilization study that's in New Hampshire, they found responses to phosphorus fertilization, and then also just this observation that there's more arbuscular mycorrhizal trees compared to ectomycorrhizal trees. And with more inorganic nutrients in the environment, such as through nitrogen deposition, that can actually shift the abundance, because AM trees tend to do better, or they're thought to do better with this inorganic nutrient economy. And we hypothesized that maybe the dominant nitrogen fixer may actually do better in under a P-limited environment, because it's historically been able to relieve its own nitrogen limitation, so that it has maybe advanced strategies to acquire phosphorus or calcium or other nutrients compared to nitrogen, whereas other temperate tree species maybe have focused their efforts on actually capturing nitrogen from the environment. And typically when we think of phosphorus pools in the soil, there's three main pools. There's mineral phosphorus, or that original bedrock that I had mentioned earlier, organic phosphorus, so that's what's coming in from decaying litter and then also microbial biomass, and then also phosphorus that's sorbed onto clay particles or iron and aluminum oxides. And so maybe, and this is just a hypothesis, there's been observations that under nitrogen fixing trees that tend to be more nitrogen rich, or their litter is more nitrogen rich, that can lead to lower soil pH because of increased nitrification that can acidify soils. And maybe this lower soil pH can actually affect the rates of that rock weathering or that bedrock weathering into soil. So plants may be able to alter their nutrient environment to some extent and actually accelerate weathering. And that's work that Sarah's group has found in Panama, for example. And maybe they utilize mycorrhizing more efficiently, or they just utilize this relationship more. And there's been seedling studies with black locusts that have shown that they're really mycorrhizal. And they also may be able to produce more phosphatase enzymes, either just due to their evolutionary history or because they've been able to acquire more nitrogen. And I'm just going to show you some of the results for phosphatase today, but basically we used an observational approach, an experimental approach to address this question, comparing black locust, the dominant nitrogen fixer, to some other temperate tree species. And to do this, Mike helped us identify 3 forest sites where we could find the 6 species that we were interested in. And then we traced these 5 routes to be able to measure phosphatase production and then also mycorrhizae. And this is Sarah and Jane in the field here at Cary, and then also Samantha, our technician, in the field as well. Fun times. And I'll just show you the results for root phosphatase activity. So we expected, again, that black locust would exhibit higher phosphatase activity rates compared to some of these other trees like black cherry, sugar maple, black birch, red oak, and white pine. And we do find slightly higher root phosphatase activity in the mature black locust trees, but mainly in the sapling trees. So they seem to really have higher phosphatase activity on a per gram of root basis. And then we also studied and compared the growth of some of these different tree species. So we added one more in for the greenhouse experiment, and that was honey locust, which is also in the Fabaceae family, so the same family as black locust. And we also fertilized with 3 levels of nitrogen that represented a range in nitrogen deposition rates. So seeing how these different trees change their phosphorus strategies in response to changing nitrogen deposition. And we were able to actually create an artificial soil environment just so we could control how much phosphorus was in the artificial soil. So we were able to create rock bags of mineral phosphorus and then organic phosphorus through litter bags. And then we also added trace amounts of phosphorus for the plants. And then this was replicated 10 times across 18 weeks. So just the 2 images here show you how much the plants really changed across 18 weeks. So they grew really, really rapidly, which I don't think I was expecting, at least maybe Sarah or Sam was, but yeah. And then we measured biomass, and we're still working on some of these other measurements such as foliar phosphorus, but root enzyme activity, I'll show you the results for. And this is on a log scale. And as you can see, black locust on the left really just has enormously high root phosphatase activity compared to some of these other species. So much higher root phosphatase activity. And it's really surprisingly not driven by growth. So here I have on the x-axis total biomass of the plants, and phosphatase is represented as just a rate per gram of root. So this rate seems to be high no matter how big the black locust seedlings are getting. And some of these other species, like red oak, for example, does seem to adjust its root phosphatase activity on a per gram basis when it grows bigger. And what's also really interesting is that this is from data from a NERC database, and that black locust has one of the highest foliar phosphorus concentrations in temperate ecosystems compared to some of these other temperate tree species. And I think there's still a ton of work to be done. We're still looking at some of these other strategies. And I think if we want to make any assumptions about the impact that black locust has on phosphorus cycling, we really have to look at phosphorus fluxes in total, but it at least has higher phosphatase activity. So it's playing some role in producing these enzymes to access and break down some of that organic phosphorus. But again, we have to look at some of these other strategies and really think about what the actual impact on the ecosystem is. Okay, so going back to that original question, will the land sink continue to take up rates of carbon at the same rates indefinitely? Well, I've shown you that nutrient concentrations alone don't indicate nutrient limitations. So even if we were to go and had the resources to collect soils across the globe, we wouldn't necessarily be able to just predict nutrient limitation alone. And that we really have to think about what's going on above ground and below ground with plant strategies. And that I think one of the first figures I showed you was that recovering tropical forests actually make up about 40% of the gross land carbon sink. So just as they recover, they're sucking up a lot of carbon. And disturbance events seem to affect forest ecosystems and nutrient fluxes, but these ecosystems tend to recover really rapidly. So they do play this really critical role in alleviating or mitigating some of that carbon in the atmosphere as they grow. But there's quite a bit of spatial heterogeneity in the tropics alone. So in the southeastern Amazon, for example, we didn't see nitrogen limitation, but we did in those recovering forests in Panama. And I think it's really critical to unravel some of that spatial heterogeneity and have some predictions on why, so we can have some predictive capacity on nutrient limitation into the future. And that plants can utilize strategies at the community level. But I think all of this leads to more questions in the future. So what are the drivers of spatial heterogeneity, and will ecosystems continue to recover rapidly if there's more disturbance events, which there are going to be in the future? And we have to start thinking about how plant communities, not just individuals, actually respond to nutrient limitation, and this can tell us a little bit more about how terrestrial ecosystems interact with soil nutrients. And we also have to think about how nutrients interact with other drivers such as herbivory and drought and pests and species invasions, because nutrient limitation is not the only thing that's going to predict, or it's not the only thing that's going to modulate how ecosystems respond to rising CO2, right? There's so many other drivers, and they're not necessarily going to respond in a linear fashion. They're going to interact. And with that, I would love to acknowledge Sarah, who is my mentor here at the Cary Institute, as well as some of my other mentors just throughout my scientific career, and such as Maya Almaraz and Fiona Soper, and then also my advisors in graduate school, Bob Howarth and Chris Neal. And some of our funding sources, such as NSF and the Millbrook Garden Club. And yeah, with that, I'll take any questions.
Thank you so much, Michelle, for a wonderful talk. We can take questions both from the audience here in the auditorium and also people who are on Zoom. If you want to put any questions in the Q&A, I will monitor that as well. Let's see, Chris.
Good talk. Really cool. I was wondering if the instrumentation forest pH experiment in Panama, where you had the hypothesis about phosphatase activity was change from the forest. It was pumps that went out, and there was like a pump for 1 meter for control. So you saw that, you kind of saw that pattern, but two nutrient manipulations away from control. Like one of them didn't do what you thought early on, the other one didn't do what we thought later on.
Mm-hmm.
What's going on in the sense of why you saw this one thought it way early?
So I'm going to repeat the question
Okay,
so everyone can hear. Yeah, I'll try. So in the experiment where you looked at phosphatase activity over forest succession, and you had predictions for how that would change with nutrients, basically you found support for some of your hypotheses, but not for others. Why do you think you didn't find support for some of your hypotheses?
Yeah, thank you so much for that question. That's a great question. It's actually up for debate a little bit. So I think like many of us, I think originally you could think that adding phosphorus, no matter what forest age, would decrease phosphatase production. And there are two reasons potentially. One is that maybe it's like phosphorus ability isn't what's regulating phosphatase production early on in succession, just because there's enough phosphorus to go around. So whatever species are there are just producing phosphatase. And the same with nitrogen. So why doesn't nitrogen stimulate phosphatase production in the older two forests? Well, maybe it's just because there's nitrogen available, and even adding nitrogen isn't going to shift the system towards phosphorus limitation because it's already phosphorus limited. But there's also some work by Zalamé et al. That's shown that different species actually have different plasticities or responses to phosphorus. So some species will really reduce the amount of phosphatase they produce in response to phosphorus, but others won't. They just continue to produce phosphatase no matter what. And it could just be that there's this species turnover that we're not really accounting for. So maybe early on in succession, we just have different species that produce or regulate phosphatase differently. And that's something that I think is really critical that we do need to understand. Thank you so much.
Peter.
So just a wonderful thought. I have a million questions, but I'll start with an old one, right? So anyway, anion exchange, that's very exciting. So but how is that playing out ecologically, do you think, to inhibit nitrogen fixation? So you have a big nitrate pool in the soil, and is that, do you think that is kind of inhibiting symbiotic nitrogen fixation, like when you apply fertilizer to a soybean field? And if so, how does that play out? Because I suspect a lot of this nitrate is pretty deep in the soil profile, and do these plants really like nitrate? And so it makes a tremendous amount of sense, but have you thought about mechanistically how that's playing?
Yeah, that's a great question. Do you want to repeat it?
I'll just repeat it. So Peter asked, in the southeastern Amazon where you studied fixation, you found that there's this huge pool of nitrate in the soil. How exactly would that play out and affect the fixation rates that you're finding, especially considering that a lot of that nitrate's really deep, and are plants liking nitrate? Can you tell us more about the mechanisms of that?
Yeah, that's a great question. And other people have brought that up before, and we don't really know, right? Because it's not that like plants might send some of their taproots, and I think a lot of the work by Nepstead et al. Has shown that plant roots, the reason why they're able to withstand so much drought in these forests that experience drought is because they send these taproots meters and meters down. We don't actually like when I was in those soil pits, I didn't necessarily see plant roots, so it's unclear if they're actually able to take up that nitrate. But even if a small fraction of it is able, it's in the surface and plants are able to take it up, then maybe at least the fine roots, because I don't think plants really send fine roots deep in the deep soils. That may explain why we're not seeing strong nitrogen fixation responses. And I think that there is this hypothesis that a lot of trees in the Fabaceae family, like the ones that are dominant in the tropics, are thought to be facultative nitrogen fixers, so that when nitrogen availability is high, they just aren't going to fix. And I think Sarah's greenhouse work has shown that. But yeah, it's really I don't know, and I would love your thoughts on if any of that nitrogen, even if it's 1%, is accessible to plants, and if plants actually experience that, because we do know that the trees that are really growing in increasing in basal area are known nitrogen fixers. So why they're not nodulating, unclear.
Maybe is the nitrate reduction going on the roots or the leaves? It's stuff I don't know much about. It's just very.
Yeah, and I think different plants can take up, like most plants take up nitrate. Some can take up ammonium and nitrate. I think the majority of them take up nitrate more than ammonium though. But other way around?
Yeah, because you have to reduce the nitrate to ammonium to use the amino acid. So there's an energetic cost of using nitrate. And so sometimes they do it in the leaves because there's more excess reductant in the leaves.
Oh,
it's interesting.
I guess we add in fertilizer experience, we add ammonium nitrates. We're adding both. I don't know if it's do you have thoughts on this?
I know. Oh, wait. I know studies in Panama have looked and like different species are taking up different forms of nitrogen. So yeah, awesome. All right, we have a question from Steve Hamilton. Can we gain insights into limitations on nitrogen fixation in tropical soils from the widespread conversion of tropical forests and savannas to nitrogen fixing soybean crops? Are limiting factors for soybean nitrogen fixation in those settings known?
Yeah, actually, that's a great question. I think that's something that even at our site, because at our site, there's actually, it's based on a farm where they're actually double cropping. So they're growing soy and corn, and they're actually still fertilizing with nitrogen. I think one of the other graduate students, Alex Adel, did want to capture nitrogen fixation rate. So the question is about accumulation from soybeans.
Yeah, and what's limiting nitrogen fixation in those crop systems?
Oh yeah, so the soybeans definitely are fixing nitrogen. And I think they're starting to, but the problem is they actually do fertilize. So it's hard to tell because they're seeing it. So the reason why we even knew anything about the extractable nitrate in the forest soils was because they were using it as a control for the cropland. So they were actually measuring nitrate leaching from fertilizer from corn into deep soil profiles, and then they use the forest as a control. I think they're finding that a lot of the nitrogen is actually accumulating. And I don't know if that they did the mass balance, if it was just due to fertilization to keep the corn crops going, or if there's also some fixed end from the soybean. I kind of believe that there was some accumulation from soybean nitrogen fixation as well. So they're not, and they add so much lime and phosphorus. They're not really constraining nitrogen fixation at all in the soybean crops. So thank you.
Other questions? Jane?
Couple questions, but the first one is for the C to N ratio. Did you look at canopy soil as well? Like I noticed that like canopy and then leaves and soil. Is that the case, or were you just looking at the canopy wood?
Yeah, that's a great question. I didn't have the litter, I think C to N ratio, so I just used canopy N as a comparison to some other sites where canopy leaves C to N had been measured. But it would be really interesting because I don't even really know what's going on, if there's a lot of canopy soil accumulation there. Because I think they're drier forests than maybe in Panama, maybe there isn't as much accumulation, but honestly, I haven't climbed the trees to see what's going on up there. So that would be really cool.
Well, and then the other thing I wanted to ask is if you have a good sense of how rapid the facultative relationship can be ramped up and ramped down. I mean, I feel like that would definitely influence in your time series plots. Also, whether or not rhizobia is a limiting resource.
So
Yeah.
can I just ask a question? So Jane is asking, how quickly can facultative fixation be ramped up and down? And then yeah,
that's a great question. And we honestly thought of, we thought of maybe there's cobalt limitation because it's also really cobalt. And maybe there's not these rhizobial bacteria around, and maybe that's why the plants can't form symbionts. So yeah, great question. I don't know. I mean, I know we've done DNA extractions and there's definitely tons of NIFH around. I don't know if they're the specific family that can colonize with root bacteria. I'm assuming there should be enough, right? They're not limited by the microbial community in the sense that there's tons of different rhizobia that can colonize roots. It's not just one species to one bacterial species to one plant species. But yeah, that's a great question.
Yeah, I guess, that's one of those first, that's a lot of being back here. One of the first broad role slide, to me would suggest that climate will scale climate
Mhm.
if you took a more granular approach and sort of soil type, would you start to would you see a correlative signature of soils on that will, are you losing the resolution because fundamentally
So going back to the original image of the productivity, how much do you think it's climate and land use history that's driving the land carbon sink versus soils? And could you, for example, take a soil map of different soil types, and how much predictive power would that have on variation in land carbon uptake?
That's a great question. Yeah. So I think the issue is, I don't know if it's the way, and I'm not a soil scientist, but the problem is like the soil orders that we have really correlate with the biomass above ground. So it's hard to disentangle the two. And I know that there's been studies that have looked at like soil type, like clays or cation exchange capacity and other soil parameters, but I don't think that they at least influence the global like carbon uptake, or that explain a lot of the variation in that. And I think with the soil order data, I think that's what you're suggesting. It's just like oxisols and ultisols are like the most weathered soils, and that's where tropical forests, which are the most productive, grow on. So yeah, but there have been a lot of studies that at least have looked at, I guess, responses of different drivers and net carbon uptake in secondary forests. And I think maybe it's just nutrient availability isn't the first thing that comes out, but it could just be the numbers that we have are so basic and don't necessarily represent like what's actually going on accessible to plants. Thank you.
Well, I think we're at 1 o'clock, so we better wrap up. But thank you so much, Michelle, for a really wonderful talk. Thank you all for coming and for being on Zoom. And let's thank Michelle one more time.